In this video, we take a look at translation, which is the process where information that came from a gene is actually used to direct the synthesis of a protein by ribosomes. And we will see that what we're looking at here is actually a piece of messenger RNA along here, and the ribosomes are these little ones that are highlighted here, and they're actually making a protein, which is our string of amino acids right here. And so what you're looking at is literally the process of translating the nucleic acid into amino acids. And so if you look at this whole process, DNA is transcribed from a nucleic acid into another nucleic acid. So you can think of it like recopying your notes. You're transcribing them. In the process of translation, you're actually shifting languages. So you're going from the language of nucleic acid to amino acid. So we want to take a look at this process and see how these ribosomes are actually assembling on the message and how they're moving along here and resulting in all of these amino acids being hooked together to give us our functional protein at the end of this process here. So if we take a look at this little thing here, what we're looking at is something that is known as the genetic code. And this is what Crick worked on after Watson and Crick worked on the structure of DNA. And if we look at this, what we see is the genetic code actually consists of three nucleotide bases that you can find in genes that are also transcribed into messages. And these three bases are known as codons, and they actually will code for the amino acids that we see in the proteins that are out there. And if you look at this little table here, we have all possible combinations of three bases in these codons with using the four different bases that we would find in our RNA molecule. So U, C, A, and G right here. And if you look at that, it gives us a total of 64 different possible combinations. Most of these are going to get used by cells, and then a few of them actually do have a very special purpose during this process of translation. And so if we look at this process here, what we can see is we just take these bases right here. And so in this case, you can see CUU actually codes for an amino acid leucine. So wherever you see this CUU in a message, the ribosome is going to eventually insert a leucine. And likewise, you can see CUC, CUA, and CUG here. These all are going to code for that amino acid leucine, along with two others over here. And so this is what's known as degeneracy. And because of this, if you have a mutation in this position right here in the codon, oftentimes it will not result in a massive change in the protein that was coded for by that gene right there. The other important codons, when we look at this, is over here, AUG. It encodes an amino acid called methionine, but this is also the start of where the ribosome is going to read the message. So this would be the equivalent to the promoter that we saw for transcription in that process. And then these three over here are what are known as the stop codons, and these would be the equivalent of the terminator sequence in the DNA that tells the RNA polymerase to go ahead and stop. These are what tells the ribosome, stop translating this message because you are done making the protein right there. And so we want to take a look at this process of translation, and similar to what we saw with transcription, we can break this process down into three phases. This first phase over here is our initiation phase, and similar to what we saw with transcription, they had a promoter that told the RNA polymerase where to start. Well, here we have this initiation that is forming, and effectively what happens is a small part of the ribosome scans along on the message, and you can sort of see that in the upper cartoon right here, and that assembles, and it's going to scan along until it runs into that start codon right there. So there is that AUG that I talked about before from our genetic code. And there is a tRNA out there that has what's known as the anticodon, and this is the complementary bases to that AUG right there. So the A complements the U complements the A right there, and then the G and the C, and these are going to form hydrogen bonds, and that will hold this tRNA in position. And notice this tRNA is carrying that methionine right there. And so this is the beginning of the process of translation. You start to form this small subunit of the ribosome, and then this tRNA carrying this methionine matches up with your AUG, and then the big subunit for the ribosome, or the large ribosomal subunit, is going to be able to assemble, and now you've got what's known as the initiation complex. All of these elements have been brought together so that you can start doing translation of your message right here. And what happens is kind of interesting right here is a second tRNA is going to be able to enter into the ribosome. And if you look at this, there's actually these little pockets that are built into it. And this pocket right over here is known as the A site, because this is where your amino acids are going to enter into that ribosome right there. So there is our second tRNA that complements this codon with its anticodon basis. So here you can see GGG is on the message, and there CCC is the complementary bases, and this tRNA is carrying an amino acid called glycine. And the next step in this process here, and the whole reason for having this ribosome, is the ribosome is known as a peptidyl transferase, and it's going to pick up this amino acid and it's going to transfer it over there, and when it does this process, it's going to make a peptide bond. And so there you can see you've just made that beginning of your protein right there. And the ribosome is just going to keep chugging along here with tRNAs coming into the A site carrying those amino acids, and then they're going to move through. And so here we can see during the elongation phase, you carrying in amino acid on a tRNA that matches the next codon right there, and then you do the peptidyl transferase. And so let's watch this right here. Now your peptide is right there on this tRNA in what is known as the P site. So A site for amino acid, P site for your peptide or your protein, and then we've got the E site, and we'll see that come into play right here. There's the peptidyl transferase, and then notice what is happening right here. Your tRNAs are going to move into what's known as the E site, and those tRNAs that come in here are empty, and so they're going to be able to exit from the ribosome right there, and then those tRNAs can float around, and they will be able to get recharged with new amino acids, and they could potentially enter back in over here if they match a codon right there. And so this ribosome with the large and small subunits is just going to keep moving along your message right there. And it pauses every 3 bases where you get a new codon. So effectively, that start codon determines where you start reading the message, and that determines your 3-letter words, which are going to get translated because they match up with the tRNAs right there. And then we come to the last phase, the termination phase. Your ribosome is chugging along, and it runs into your stop codon right there. And the interesting thing about this is there are no tRNAs that match up with your stop codon. So instead, what's known as a release factor can enter in the A site, and this is going to cause that ribosome to basically pick up your protein, and it tries to move it over, but there's no amino acid in that A site. So the ribosome can let go of your protein, and that's your long chain of all those amino acids, and then this tRNA can fall off. The large and the small subunits are going to fall off of your message, and your message can get reused. And here you can see there is your large and small subunits, and then those parts of the ribosome can float around. They could reassemble on this message and start the whole process over. They could go to another message and start doing translation, because these ribosome elements are reusable, just like your tRNAs are reusable in order to make new proteins that are out there. So hopefully this process helps you to understand how we can look at the different stages of translation and see how these ribosomes are playing a role in this process of taking that information from a gene, turning it into a message, transcription, and then taking that transcript, your message, and the ribosome is going to translate that into amino acids. And remember, these proteins can fold up and give you that 3-dimensional structure that's going to have an effect in the cell. Whether it's as an enzyme or a structural component or something that's secreted really depends on what genetic information was in the gene, and that determines the final sequence of amino acids in our proteins, and those can fold by themselves.